Hello, my name is Sean Crafts, and I'll be presenting my experiment crossing Drosophilidae, male brown scarlet, and female sepia. The purpose of my experiment was to use quantitative data from phenotypes in two Drosophila generations to investigate the genotypic relationship between the genes. Drosophilidae served as a good model organism because of its rapid maturity and reproduction rate, with a 7 to 10 day maturity cycle and 20 to 30 day lifespan. They have a low cost in maintenance. The phenotypes are easily identifiable, and their genotypes contain many human disease-causing genes. My initial hypothesis was if a female curly plum fly is crossed with a male brown scarlet, the F1 generation will have wild-type fuzzy legs, plum eyes, and curly wings. However, a bacterial infection killed off that generation, and I had to make a new cross with male brown scarlet and female sepia. My hypothesis for the second cross was if I cross brown scarlet with sepia, and sepia is a recessive autosomal gene, then the offspring in the F1 generation will be 100% wild type, and the F2 generation will be 75% white-eyed and 25% reddish brown. For my methods, I first extracted virgin female sepia flies and male brown scarlet using FINAP. I transferred approximately 6 sex pairs of flies to a vial labeled F1 generation. 7 to 10 days later, I removed the P1 from that culture to prevent crossbreeding. After the F1 matured, I transferred 6 sex pairs of flies to a vial labeled F2 generation, and then 7 to 10 days later, removed the F1 from that culture. Finally, I recorded the number of progeny for each phenotype. My expected fly generation data was 100% heterozygous wild type for the F1 generation, and for the F2, 50% heterozygous wild sepia, 25% homozygous wild, and 25% homozygous sepia. And here is a test cross showing how I came up with those ratios. For my results, I had 4 different phenotypes separated into 7 to 2.5 to 3.3 to 1 distribution ratio for red, orange, brown, and white phenotypes. And my data did not support a sex link because there was a fairly equal number of sex distribution for each eye type. For my conclusion, my second hypothesis was falsified. Production of 4 different phenotypes, red, orange, brown, and white eye colored, falsified my second hypothesis, proving sepia is not an autosomal recessive gene with an expected F2 offspring ratio of 75% white eye to 25% red scarlet. This may have been because the brown scarlet had a gene interaction on a different chromosome resulting in the additional phenotypes, or perhaps both sepia and red scarlet phenotypes could be autosomal recessive due to the fact that we saw all 4 colors on each species in a ratio not far from 9 to 3 to 3 to 1 that we would expect in the F2 of the autosomal dihybrid cross. I would just like to thank Dr. Hanna for collecting and preparing the F2 generation in the second test cross. And there are my references.